Hello students, today we are going to discuss the topic sexual reproduction in angiosperms, in which Megasporogenesis we are going to discuss. That is the process by which the flowering plants or angiosperms, they form the megaspore. The term Megasporogenesis itself means that it is the genesis, means the formation of megaspore. So in these plants, as you know, that there is a process of sporic meiosis, a meiosis by which they form the spores, which are part of the sexual reproduction, microspore for the males and the megaspore for the females. Now, how is this megasporogenesis, that is formation of megaspore, is going to take place? As you see in the diagram, that this particular diagram that you see out here, now this is actually ovule, because you can see the nucellus here. Now nucellus outside, this is the tissue which is actually not outside, it's inside only, inside the ovule, but this is the tissue which is sporophytic and nutritive in nature, diploid in nature, and this tissue of only one particular cell, which is called a megaspore mother cell, will arise from this tissue and is going to give rise to the megaspore tetrad, which you can see in this diagram, with the help of meiosis. Now this meiosis would be called as the megasporogenesis, will result into the formation of megaspore tetrad. Now what we need to understand is that this megaspore mother cell, which is actually near the micropyle, so this actually arises at the micropyle, it is separated out by a covering around it, which is called callus. It gets surrounded by a special covering of callus, which is a glucan in nature, and is ready for meiosis. It is a prominent, distinct nucleus, dense cytoplasm. It's a cell which is now ready for meiosis. Meiosis, the reduction division, will result into always 4 cells. Now here these 4 cells are megaspore. These are megaspore tetrad. So this is a megaspore tetrad because there are 4 cells. As you can see, these are 1, 2, 3, 4. Now these 4 cells, which are arranged in a linear order, this tetrad is linear because there can be different types of tetrad shapes, but in megaspore, this is linear tetrad. And the nucellus is still outside. It is visible here in the diagram. Now towards the chalaza, a bigger cell of megaspore is visible. And this is the one which is near inside the nucellar tissue, and it is able to derive more nutrition. So this is the only one which is going to survive in most of the cases, and the other 3 towards a micropyle are going to degenerate. If only this one functional megaspore will survive, only 1 out of 4 will survive, then the further development of the embryo sac or the female gametophyte would be called as monosporic. So the most common which we see is monosporic. So here we will consider this, that this only one has survived. Also, you have to take a notice of this, that since it is meiosis, so the megaspore formed is haploid, and haploid in the plants, in angiosperms or in higher plants, it is a gametophyte. Since it is a heterospory, the microspore was supposed to give rise to the male gametophyte, and the megaspore is supposed to give rise to the female gametophyte, which is also called as embryo sac. So we'll further down, we can see the diagram of embryo sac. Now monosporic embryo sac, I'm repeating again, what is this? This is when out of 4, only 1 megaspore has survived, and this functional megaspore will form the embryo sac or the female gametophyte, and the process is called megagametogenesis. What is the process called? It's megagametogenesis. Fine. Now, in the megagametogenesis, there is a series of mitosis. As you can see, the first mitosis out here, that is two nuclei are formed, and the second mitosis, first 2 nuclei, and then second there are 4 nuclei, and the third there are 8 nuclei. These nuclei which are formed, this is simply free nuclear divisions. Immediately there is no cytokinesis or wall formation. So this is free nuclear divisions. So 2, then 4 nuclei, and then finally 8 nuclei are visible here. Free nuclear divisions. Now, what happens to these free nuclei? Slowly, as you can see here, there is a development of the cells. So cells have, that is cytokinesis have taken place and the cells are developing. There are 3 cells which are visible here at the micropyle. M stands for here micropyle, and the C here stands for the chalaza. This is M for micropyle, C for chalaza. Look at the micropylar region. There are 3 cells which I have named as egg apparatus. I have labeled. There is a cell out here which is central cell. We'll see more details here in the next diagram. And in this central cell somehow is lacking one cytokinesis, due to which there are 2 polar nuclei. These are 2 nuclei here and they have not undergone a cytokinesis. Whereas at the chalaza, there are again 3 cells, and these 3 cells are called antipodals. This arrangement is often called as a 3 + 2 + 3. In terms of nuclei, 3 towards the micropyle, 2 in the center, and 3 at the chalaza. So this development of nuclei or arrangement of nuclei is called as 3 + 2 + 3. Now this gametogenesis continued, and you can see there is a full-fledged, mature embryo sac diagram. So it is very much in your textbooks. This is the mature embryo sac, you can see. Now at the chalaza, there are 3 antipodals, which we have seen here also. So now the only thing is the chalaza orientation is this side. So see the antipodals here. And central, there is a big cell, and this is called as central cell. We have already seen, and the central cell has these 2 polar nuclei. Okay. And so this has cytoplasm, of course. And then you can see at micropyle, there is egg apparatus, and egg apparatus is having which cells? There are 2 synergids right at the micropyle, and there is 1 egg behind, and it's the largest, it is among the egg apparatus, it's the largest cell. It has a very distinct prominent nucleus, but the synergids are having a special filiform apparatus which is visible here. So these are the finger-like structures like this. So this is called the filiform apparatus. The functions of all these cells are different. Antipodals, not much functions known, but the rest of the cells, they have functions like this polar nuclei which are present in the central cell will take part later in the double fertilization in triple fusion, that is specially called a triple fusion. And in triple fusion, these 2 polar nuclei, just before fertilization, they fuse to form a secondary nucleus which is diploid. A secondary nucleus which is diploid. And with that, the male gamete fuses. So we'll come to that later, but initially, we have to understand one thing, that the pollen tube will enter through the micropyle. That is the most common way of entry, which is called as porogamy. Most common way of entry of the pollen tube is through the micropyle. Now, when it enters, it finds one of the synergids is degenerated. Whichever is degenerating on that synergid, this particular pollen tube that we are talking about, this pollen tube will burst out and it will discharge its contents. So therefore, synergids will serve as seat for pollen tube discharge. So what is the function of synergid? That it is a seat for pollen tube discharge. It's one of the one of the functions. Then the second function, even before that, in fact, in sequence, that is even before, that synergids, especially these filiform apparatus, they were secreting some chemicals. What for? These chemicals were for to attract the pollen tube. because pollen tube is traveling right from the stigma through the style. So it's a long distance it has traveled, and it is finally reaching the microbial of the ovule. It has to locate it. So for location, there is a chemotaxis. It's a locomotory, it's a chemotropism. It's a chemotropic movement. Why we call it a chemotropic? Because this is not locomotory, unlike the lower plants, bryophytes, pteridophytes, where the male gametes, anthrozoids, were motile. They were moving with the help of water. Even they were attracted by the produced by the archegonia. But in this case, there is no archegonia, there's no water requirement for the fertilization. So this is a growth of the pollen tube. Since it is a growth, so it is called chemotropic. So it's a chemotropic attraction and movement of the pollen tube towards the micropyle. So it discharges its content on one of the synergids, and then first male gamete will fuse with the egg and will result into a zygote, diploid zygote. So that is the syngamy or fertilization. The second male gamete will swim inside, it will reach inside, and the second will fuse with this secondary nucleus, which I was telling just now. That secondary nucleus is formed by the fusion of the polar nuclei, right? So this is the summary of the megagametogenesis and the embryo sac. We'll soon continue with another video for double fertilization. Thank you.